Hi, my name is Alina and I'm glad to see you on my channel. I'm a student and I'm studying in MSU for immunologist. And now I decided to start a circle of lessons about cell biology and molecular biology. I want to apologize for my bad English. I'm doing my first steps in teaching in English, so if something will be not so clear, you can write down it in comments and I will try to explain everything. Okay, let's start. Today we will talk about epithelium tissue. Yes, we start the circle of histology. And first of all, we should say that epithelium tissue is a layer of cells. These cells have tight contacts between each other, and the extracellular space and extracellular area almost absent. Epithelium tissue is a barrier, so it is organized to avoid any penetrations. Yes, they have some exceptions, but we'll discuss it later. Next feature is basement membrane. We can imagine it like a film that divides epithelium tissue from underlying connective tissue. Basement membrane has a complicated organization. It is a network made of enormous quantity of different proteins, and we will discuss only the most key of them. Another meaningful characteristic is the polarity, and it is connected with the basement membrane. The basal and the apical part of cells have different structure, and there are different proteins on it. For example, tight junctions near the apical part of cells and desmosomes near the basal part of cells. So, all of these contacts are absolutely vital for cell. It holds her on the basement membrane via hemidesmosomes. Side contacts maintain exchange of information and security, and moreover, tight contacts have a significant meaning in our immune system. It provides our first barrier. Pathogens with difficulty make their way through epidermis because there are a little space between cells in our epidermis. Next point is absence of vessel. This is widespread mistake that in epithelium tissue there are some vessels. No, it's not true. All vessels are localized in connective tissue, and so epithelium tissue consume nutrients from the underlying connective tissue, and with help of diffusion it spread to the higher layers of cells. Next point is keratin. It is a very important protein. Keratin are markers of epithelium tissue. So we can have a lot of exceptions, like pancreas, where is no basement membrane and vessels penetrate the organ, but how can we say that pancreas is made of epithelium tissue? Via molecular markers. Every type of epithelium tissue contain any or some of keratin types, and we will discuss them in the particular types of epithelium tissue. But there is the opposite situation when we have three types of cells that are not included into epithelium tissues, but they have basement membrane, for example. These are adipocytes, neural cells, muscle cells. All of them have basement membrane, but they are not epithelium tissue. How can we identify it? Via the molecular markers, via keratins. So yes, it is very important proteins in identifying. And the last point is truly important. It is the ability to regenerate. I think that I will make a separate video about the ability of regeneration, and if it is interesting for you, please write down in the comments and I will do my best. And so I want to discuss each property separately. First of all, basement membrane. As I said, it is a separator between epithelium tissue and underlying connective tissue. It regulates transport between vessels in connective tissue and epithelium cells. It contains two parts, basal lamina and reticular lamina. Now, this is the beginning of the most hard part of our lesson.
proteins. There are enormous proteins in basement membrane, but we'll discuss only some of them. So the structure forming protein in reticular lamina is collagen type 3. It is organized into collagen fibers, and these fibers are jointed to a basal lamina through collagen type 7. Yes, we can see it on the picture. And the main protein of basal lamina is collagen type 4. It is organized into complicated network, not fibers, exactly into network, and it is stabilized with the help of collagen 5 and collagen 7. So on the picture we can see that the reticular lamina is near the connective tissue and the basal lamina localized near the epithelium tissue. So let's observe how cells connect with basal lamina. First protein group is proteoglycans. They are attached to collagen type 3. One of them is perlecan, the most common protein in the basement membrane. So nidogens are another group, interesting group of proteins that connects laminins with proteoglycans. So laminins connect nidogens with integrins, and integrins are the very important transmembrane proteins. So integrins, it is the surface of the cell, and we have a complicated chain, and we have a chain like we have collagen type 4 that is connected with proteoglycans. Proteoglycans is connected with nidogens, and nidogens is connected with laminin, and laminin connects with integrin that are transmembrane proteins. Yes, we have a really difficult chain of proteins that maintain the link between cell and extracellular space and the basement membrane. So why it is so complicated, why it is so important? If cell disconnect with the basement membrane, it can lose its stem characteristics and it can go into differentiation. Yes, we will discuss the topic of stem cells and differentiation in the next lessons, and in the next lesson we will be talking about classification of epithelial tissue and localization of some types of them. So if this lesson was useful for you, please write a comment and subscribe, and I will be glad to see you on my channel again. Bye bye.